introduction of you, if it is okay.
Sure.
Thank you. Hi everyone. So here our first speaker, Dr. Jingshing Wu, is an assistant professor in the Division of Neonatology and Newborn Nursery here at UW. And she's also a physician scientist focused on how environmental factors influence health. As a developmental biologist, her research explores the molecular mechanisms by which environmental exposures may contribute to or protect against congenital heart disease, with the goal of informing public health strategies and developing preventive and therapeutic approaches. With that, I'm going to pass it to Dr. Jingxing Wu for her presentation.
Thank you for that. Again, I'm apologizing because I can't get out of the presenter view when I try to share my slides. Does anybody have any technical advice for me? Seems a little more straightforward on Zoom.
Maybe try sharing it and see what we see, actually, because sometimes what you see on what's shared is the actual presenter mode or the full mode, whereas
Okay.
you on your just try it
You
and
can
see.
try share the whole screen
Yeah.
instead of sharing a window.
Like, what do you guys see right now?
That's your look good, actually. We don't see your notes and we just see the main slide.
Yes, now it's good. Yeah.
Oh, okay. Okay. Well, that's really confusing.
I know. It's Teams. Come on. What you expect?
Yeah. Okay. Well, thank you, everybody, and thank you for that wonderful introduction. I'm really honored to be able to present some of the work that we are developing in my lab as efforts to understand prematurity, birth, and then kind of implications that we do as neonatologists on the developing heart, and then also the sort of effects on long-term cardiovascular outcomes. And so the title of my talk, Preterm Birth is a Cardiovascular Disease Risk Factor. And then our lab's efforts are focused on building translational models to try to understand how preterm birth is indeed a risk factor for cardiovascular disease. So for many of us who take care of children who are born either premature or have congenital heart disease, they're essentially having prolonged hospitalizations that include a lot of interventions, including mechanical ventilations, high oxygen exposures, medications, nutrition, either deficiencies or perhaps overcompensations, and then also, cardiovascular collapses that in effect can affect cardiac perfusion or can affect perfusion to the entire body. And all of this has lifelong consequences, as many of us are aware, including, oxidative stress, inflammation, and we have evidence of disease even during this neonatal period, in terms of vascular alterations that continue to present clinically as evidence of insulin resistance during early infancy, and then later on in childhood, resulting in cardiac alterations that is even detectable by echoes, especially contractility deficiencies. And then in adolescence and adulthood, then you start to see presentations of a lot of what we consider adult onset cardiovascular diseases. These diseases are happening at much, much earlier ages in individuals who are born premature, including hypertension, strokes, myocardial infarctions, metabolic syndrome. And not only are they presenting earlier in life, these diseases are also presenting more severely. And so people who are born premature are have up to 50% increased risk of dying earlier from these cardiovascular diseases. So I think, this is definitely something that I feel is underappreciated. especially, premature birth doesn't even make it into our medical records for the most part, or it doesn't stay on as a part of our medical record into adulthood. And in the countries who do keep track of some of these longer term outcomes and have more complete medical records, there's a lot of evidence that premature birth can affect, multiple different organ systems. But a cardiovascular system is being one of the major ones that can lead to early diseases. In addition, we have data showing that basically just being born premature, even in someone who didn't necessarily require a lot of interventions during that neonatal period, sort of your late preterm infants who are sort of they were born 34 weeks, they maybe needed some CPAP or just mostly gavage feeds. And sort of our kind of standard feeder growers, those kids too are still at risk for developing these cardiovascular diseases later in age. So, not only from a public health standpoint, I think it's worth having conversations with our adult colleagues, what are the primary care interventions that should be more consistently performed, but also, our pediatric colleagues, that to be more consistent with blood pressure monitoring and counseling, children, before they reach those adolescent stages where they might be at risk for high risk behaviors that, hey, because of your premature birth, that it puts you at higher risk for these cardiovascular diseases. So, smoking and engaging in high risk behaviors is going to also have a higher impact on their health later on. So in the meanwhile, I think, definitely we need to focus on our efforts from a public health standpoint, but from a medical standpoint, while we have this issue that premature birth increases the risk for cardiovascular disease, we have a urgent need of developing therapeutics that decrease this risk for, our most vulnerable patients. However, we really have a very, very basic understanding of the sort of underlying biological mechanisms that get disrupted. And so that's why, if being a neonatologist, it ends up being a personal, very motivating clinical question to pursue here in the lab, to ask the question of, why does premature birth lead to long-term cardiovascular disease? And we have some information, and a lot of that information just comes from the sort of biological understanding that during the later stages of cardiovascular development, the heart begins to prepare itself for the higher ATP demand that occurs once the baby is born. So in utero, there's a low ATP production, and the heart primarily uses glucose for its energy source, but then the heart needs to be able to make that transition to the exuteral environment where energy demands are much, much higher, and transition to fatty acid metabolism. And so we have in our lab some mouse studies where we've looked at both at the transcriptional level, so looking at RNA sequencing as well as protein expression, that a lot of the proteins and genes that regulate these processes for ATP production start to increase during the later stages of cardiac development, at least within the mice. And so proteins and genes that are involved in oxidative phosphorylation, oxidative stress, glucose metabolism, and fatty acid metabolism all increase. In preparation for cardiac maturation that happens during the neonatal period, once the baby is born, to help the heart prepare for aspects of cell growth, mitochondrial proliferation, structural changes at the sarcomeric level, as well as make that glucose to fatty acid metabolism change to essentially accommodate for that increased ATP production. And then we also have evidence that in individuals who are born premature, that their ability to combat oxidative stress is significantly lower compared to those who are born at term. And you can see that from the study here, where individuals who are born premature, that there's a pretty, direct correlation between gestational age and their ability to have their antioxidant capacity. And so the hypothesis that I have been generating and working with essentially is essentially looking at how what are the developmental processes that happen at the later stages of cardiac development that premature birth ends up disrupting, and then how does the postnatal adaptation get disrupted because of that early interruption by premature birth. And so we have a couple different models to study this question and to test this hypothesis that premature birth interrupts the later stages of cardiac development and prohibits the heart from being able to adequately mount a appropriate cardiac maturation transition. Human studies is always the more ideal way to perform any study. However, all of us on this call are very much aware that cardiac tissue is notoriously difficult to access, especially in those who have a very, very high chance of survival, and in anyone who we do have access to their tissue, in a biobank, for example. You have to take into context of why the person ended up having the demise, what were all of the cardiovascular and hemodynamic potential effects that that person encountered, and, the medication toxicities, et cetera, infection included, that would affect our ability to really interpret our findings. And then the other part of the problem is that we don't really have extensive biobanks that have tissue from individuals who are born premature. Our biobanks actually we have, many more biological samples, especially, thanks to efforts by the PCGC. And, I know that there's a good biobank at Medical College of Wisconsin for individuals with congenital heart disease, but we haven't really sort of tapped into this opportunity for our infants who are born premature. And so kind of our next best approach would be to use human inducible pluripotent stem cells to differentiate them into cardiomyocytes. However, one of the major limitations to using this as a model is because differentiated cardiomyocytes, they're very, very limited in terms of their ability to become more mature cells. The cellular phenotype is a lot more fetal. And so while it may be helpful for me to study, what is going on during fetal stages of cardiac development, as one of my key questions of cardiac maturation, this would be a limiting factor. And so this is why I have turned to using animal models. Mice, I showed you some earlier findings from our mice. Well, as you can imagine, there are a couple limitations to using mice. One is mice, actually, when they're born with approximately sort of the equivalent of humans at 20 some weeks gestation, sort of physiology, including their lungs are a lot more, they're saccular, they're not alveolar, they haven't alveolized, and their hearts are sort of are much more transcriptionally and histologically closer in approximation to humans at 20 weeks. And then on the other hand, mice don't develop bronchopulmonary dysplasia. They don't have, a lot of the complications that humans have at these gestational ages. So they're really an imperfect model. Mice do develop bronchopulmonary dysplasia if they're exposed to high oxygen levels. And so there's specific manipulations that we would have to do in order to really mimic the effects of prematurity a little bit more profoundly. And so therefore, we also turn to other larger animal models, pigs and sheep being kind of the primary models that we have used within the neonatology world to study prematurity. There's a couple different groups actually around the world that use pigs as an animal model. The major difference between pigs and sheep as animal models is that sheep tend to have essentially one animal per birth, which on the one hand is a lot more closer to an approximation to human physiology, but on the other hand, from a sample size standpoint, these are costly experiments to perform to begin with. And so therefore, pigs, 1 litter tends to be anywhere from 5 to 6 up to 8 to 9 would be able to allow us to have, some additional biological replicates to expand our findings a little bit more. And then the other nice advantage that we have here at UW is the Wisconsin miniature swine. And so the kind of pink pigs that I showed you earlier, those pigs, we typically call them conventional pigs. They're, I think, specifically also referred to as Yorkshire pigs. Those are the pigs that you see basically at the supermarket. They're bred for agricultural, for human consumption. And so those pigs are less, they weren't designed for research purposes. And so therefore they were bred to be leaner animals and not acquire fat in the same way. Whereas the Wisconsin miniature swine actually, develop obesity. We actually have several metabolic disease models that we can use in the future. And Wisconsin miniature swine are also developed specifically for research purposes, giving us the advantage of being able to understand really at the metabolic level what are some of the physiologic changes, and then prospectively looking at, what are potential maternal factors that can then subsequently affect fetal development and fetal health outcomes. And so as our approaches in the lab here, the environmental factors that we're currently introducing are really high oxygen. And so we're developing a model where we can take these piglets, deliver them prematurely, be able to have interventions anywhere from mechanical interventions, mechanical ventilation interventions to providing medications, resuscitation, as well as different nutritional adaptations to promote their growth. But our the primary factor that we're looking at the moment is oxygen. And then we're going to look at them from both a transcriptional level using ATAC-seq and RNA sequencing technologies, as well as at the proteomic level, looking at gene expression or protein expression, and post-translation modifications. And then finally, there are many opportunities here to look at functional assays such as calcium handling, contractility of individual cells, and also mitochondrial metabolic assays using seahorse. So I'm really, really excited to develop this model and use all of these different molecular approaches to dissect out, what are some of these biological pathways that are disrupted, and then to use that information to really try to address this problem, therapeutically. And, I would love to, sort of take this opportunity as I'm sharing this information with all of you to also sort of hear your thoughts on model development, on collaboration opportunities, on different approaches that we could integrate into, studying this disease and other disease models that any of you would be interested in pursuing. And so, additional future directions for this project, in addition to using some of the research techniques I described, I do think that there is a huge opportunity here for extensive collaborations, within our community, and to leverage these findings towards developing, future research directions and therapeutics. And then, I have many people to thank for these research efforts, including members of my very young and developing lab. Our lab is about a year old, so I'm pretty excited that we have gotten ourselves to the point where we have been able to start developing this premature piglet model that also wouldn't have happened without financial support from the Meritor Foundation, and then as well as additional members and mentors, including Carter, who's been really instrumental in helping my transition and launching of the lab. Eric Schmuck is our Director of Research Operations here at the Center for Biomedical Swine Research and Innovation. And then Ying Ge is a professor and cardiovascular proteomics expert who will be helping us with the proteomics experiments. So here I have a few more minutes to take any questions or any thoughts.
Thank you, Mrs. Joy. We actually have a large group here. So Ganesh Kanduri, James Afolian, the Akiko Mamotos, we have a lot of animal models for the bronchodysplasia. So they're using the sheep model, the lamb model for that. They're doing a surgical model, and they have some genetic models as well. I don't know if you've been in touch with them.
I have not personally. I have, heard of their work and been following the work. It's, it's always one of those tricky points. And I definitely think that there definitely, collaboration opportunities there. One of the things that, I think I have to think about a little bit from a technical standpoint is that some of the assays that we perform have to rely on fresh tissues. And so then there's a sort of additional element with, how do you transport tissues and make sure that they're, going to be able to survive the distance from Milwaukee to Madison.
Yeah, and we can collect things for you if it's helpful. I mean, I just think it could be an opportunity just to explore some of that might even validate what you're doing there.
Yeah. I definitely think, sheep pig validation is also, pretty significant. One thing that I also even just like looking at the literature, there's no cardiac atlas out there, even just for the later stages of development. Because, as you can imagine, humans, sort of all of our samples kind of end at the 24-week, 25-week point. So it's a pretty big gap. And then, animal models are sort of imprecise for all of their own reasons.
Yeah. Thanks.
Yeah, Jingshing, I just want to emphasize, I think you're off to a great start. I love this. And I like the fact you're kind of using different model systems to get at these questions, because I think they'll be nice to validate each other. As you were mentioning really well, I think each have advantages and each have absolute disadvantages. So I think by coming at this in a multi-pronged approach, you're going to make some decent progress. So nicely done.
This is a challenging, but also like very innovative model to use this pig. So I'm wondering, like, after you induce them prematurely, could you also isolate like the pig cells and then do some cellular level experiments?
Yeah, for sure. We actually, didn't list all the collaborators, but we actually have one collaborator who can actually look at sort of mice and mechanics. Dan is his first name. I can't remember what his last name is. I apologize. I think he's out at Utah. He's out west somewhere. And so, and then there's, and I apologize. I actually probably should have also listed Tim Camp here. Tim Camp's lab, actually, they are able to look at a lot of those functional assays I described with calcium handling. And then, obviously Carter's lab, is able to, using engineered heart tissues, is also able to look at contractility with, differentiated cardiomyocytes.
So do you think there's a lung cardiac access signaling here at all?
Oh, I'm sure there's a multi-organ access, I am sure, and the poor kidneys, we tend to forget about them, but they definitely, I would be surprised if, they weren't actually, metabolically involved somehow. So,
yeah. And what happens if you just put primary cells under hypoxia? How much does that recapitulate the environment?
Yeah, so that is, I didn't have time to go through all those
simplistically.
Yeah, we concurrently actually do have stem cells that are growing that we're trying to differentiate them into that we're trying to differentiate them under low oxygen conditions. So this would be 5% oxygen. Yeah, they grow very slowly, and so we are right now in the throes of trying to figure out, like seeding densities and how much chur to put on and all of that. So maybe in a year or so I can be able to tell you what that protocol is. But yeah.
Right.
If you think differentiating cardiomyocytes in 21% oxygen, by itself can be tricky. 5% is its own beast.
I really enjoyed your talk. It's Jeanette Strasburger. I just wanted to highlight one thing you mentioned, which is that the prematurity does not show up in the electronic medical records of the adult. And I think, that is one area I've been interested in too, because we have trouble with finding fetal heart rates in the electronic medical record retrospectively, and doing any kind of retrospective research in that. And I think that we do need to start working with some of our organizations to begin to insist that our medical record companies create algorithms that allow us to better track things that are really critically important, like for us, fetal heart rate, or for you, the presence of prematurity in an adult. I know Rainer Gedeit and some of the people at MCW have been working with EPIC, and I think EPIC is, being in Madison, it's an opportunity for people who are interested in medical record research to really work in that as well. So I just wanted to make that comment, but I really enjoyed your talk.
Thank you so much. Yeah, well, there's medical record is one thing, and then the other thing is getting our, pediatricians and our adult primary care physicians on board with valuing this as a risk factor. I think a lot of people just kind of poo-poo it. It's like, oh, well, you seem normal, and you're, seem to be very highly functional, and you're exercising and eating well. What else can we do?
Yeah, I've seen it as included on the problem list early on, right? But then as kids get older, it falls off. Like somebody deletes it or removes it because it's a resolved problem. And that's the question, if with the EMR, if you can go back and, search for what were old problems that may have been deleted, which I'm sure it retains somewhere.
You're right, Carter. And some of that is really what they consider to be a fundamental vital sign or a fundamental application within the medical record. So I think that's where we have to begin is having some of this not be able to be dropped off of the medical record.
Totally agree.
Hi, Dr. Wu. This is Aoi Mitchell. Thank you for your talk. I really enjoyed it as well. Please know we would be really excited to help you explore how the biobanks here could help validate and enable your efforts.
Thank you so much.
Thank you.
Yeah, I think long-term, Carter and I actually have also discussed this, is that, it would be wonderful if we could just pool resources together, especially with the Forward Alliance.
Exactly.
Good plug for it. Thanks.
Well, if anyone is interested in collaborating and has other research ideas, please reach out to me. My email is down here. Okay, thank you.
Thank you. And before Lou starts, I just want to acknowledge Siki Lee, who just received an AHA postdoctoral fellowship notification yesterday. So congratulations to Siki and her mentor Lou as well. So great news on that front. And for people that are on campus, there's cupcakes outside my office.
Thank you very much, Joy. So Suki just got the notification a couple of days ago. So actually today I'm going to talk about mostly of her data today. And I feel, could you be able to see my slide?
Yes.
Shared?
Yes.
Yep.
Okay.
Yep. Looks
Okay.
good.
I'll just move away this one. Okay. All right. Now seems better. Okay, so today seems like cardamom maturation sessions. So today I'd like to talk about this centrosome dynamics and how it plays a role in the cardamom proliferation and differentiation, and how this is relevant to the heart development and disease. So division and maturation are largely mutually exclusive, like in mammalian cardiomyocytes. One, during the postnatal stage, the cardiomyocytes, they started to exit the cell cycle and started to undergo extensive maturation from transcriptional level, metabolism, structural, and electrophysiology. And for this project, we are very interested in understanding what kind of cellular mechanism really coordinate this cell cycle exit and functional maturation. And in this project, we are looking at centrosome. Centrosome is a non-membranous organelle, and it is very well known for its function in organizing mitotic spindle when the cells go through the cell, the mitosis. And here it is composed of a pair of centriole and surrounded by this protein-dense, the perinuclear pericentriolar material. So usually it has up to hundreds of proteins, and it forms this scaffold that help organize the mitotic spindle. What is less well known is during the terminal differentiation processes, including in the neuron cells, myotubes, and cardiomyocytes, you will see the centrosome they started to disassemble, and with part of the pericentriolar material, they started to redistribute into other organelles or different cellular locations. So what it happens is the microtubule gonna reorganize from different places into the cells. So back to about a decade ago, and some research from Dr. Felix Engel's lab, who's also in the heart regeneration field, they used this red ventricle cardiomyocytes, and they found that in the embryonic stage, this centrosome started still show this intact structure. But however, during the postnatal development, two of the pericentriolar material called matrix, one is PCM1, another one is PCNT. They started to translocate into the nuclear envelope. However, the functional significance and what is the underlying mechanism of this whole process is still unknown. So in our opinion, we believe that this process is not only a passive process during the cardiomyocyte maturation. So therefore, we have this hypothesis that two distinct modes of microtubule organization, one through the centrosome, another one is the non-centrosome structure, governs the transition from proliferative to the mature cardiomyocyte states.
Does it stay in the envelope after in the adult?
Yes. Yes. So by the way, I think Joy mentioned a very important part. So this PCM1 stays on the nuclear envelope like sustained until the adult stage. So what it happens is in the field for the past 10 years, and people started to use PCM1 as a nuclear marker for cardiomyocytes. However, the function is unknown. So with that, Siki has been leading this project was the visiting graduate student before, and right now is the first year postdoc fellows. And we started to use human iPSC-induced cardiomyocytes to understand centrosome disassemble. And usually the iPSC-derived cardiomyocytes by day 8 to about day 10, they started to like beating, but then gradually they will develop a certain degree of maturity, although it's not as mature as the primary adult cardiomyocytes. So based on their mature level, we define them as in the early stage, usually before the day 20, and some middle stage, we calculate for about 1 to 2 months, and some to a little bit later stages. And this is also based on published results from the single cell transcriptomic changes. And so with that, we started to define the centrosomes and mostly focusing on two of the proteins. One is PCM1, another one is PCNT. So as you can see from the early stage in those iPSC cardiomyocytes, this PCM1 showed this centrosome typical structure. And PCNT shows same thing. So that indicating that at this stage, the cardinals still possess this intact centrosome structure, which is also consistent with their proliferative capacity. And gradually, you will see the disassemble of the PCM1 over here diffused into the cytosol, and gradually they fully surround the nuclear envelope. And PCM1, you will see it has a slower disassemble compared to PCM1, and it also showed a lower degree surrounding the nuclear envelope compared to the PCM1. So which is suggesting that this centrosome disassemble is actually a stepwise process that may also corresponding to the maturity level inside of the cardinocytes.
Do they ever physically interact, the proteins?
Like PCM1, PCMP, they do.
Okay, so
Yeah.
earlier, clearly.
Yes.
Okay.
Yes. When they are on the intact centrosome.
Okay.
Yes. And so because centrosome is famous for its function in organizing the microtubule, so therefore we started to look at the microtubule organization with the alpha tubulin. So in the early stage, again, this typical like centrosome organized the microtubule, like this radial structure. But gradually when it's getting to the middle stage, you could appreciate that the microtubule started to really surround the nuclear envelope. And you will also start to see the signs of this parallel microtubule structure. And when you really look at the overlay, you could see like it also aligned very well with the sarcomere structure over here. So when it's getting to the adult stage, if you see the primary adult cardiomyocytes, they actually very parallel from the longitudinal level and like aligned very well with the sarcomere. And in order to understand whether PCM1 play a role in that, and we, with the CRISPR-Cas9, knock out PCM1 in the human iPSC cells, here just a validation that PCM1 is knocked out successfully. And then we start to differentiate the cells into the cardiomyocytes, which a little bit to our surprise, and here you can see from the contractility that both the control and the PCM1 knockout showed very nice contractility. And even very often we see that PCM1 knockout cardiomyocytes at this later time point showed very good sarcomere structure, even sometimes better than the control condition. And then we continue culture those cells to middle stage to the later stage. And what we found is in the control, as we all know, like those sarcomere started to gradually mature like over time. But however, in this PCM1 knockout, you will see the fully like disruptor of the sarcomere structures from the middle to the later stage. When it really after the 2 months, those cardiomyocytes completely lost the sarcomere and they become large and they cannot beat. So in addition to that, we also examine this troponin I3, which is a mature cardiomyocyte marker. It also showed a reduced level with the PCM1 knockout over time. So which suggesting that PCM1 regulate the sarcomere structural maturation and organization. So again, when we look at the microtubule, we found that in the control condition, microtubule started to also mature and surround around the nuclear envelope. But in this PCM1 knockout, we saw a reduced number of the microtubule, but most drastically is around the nuclear envelope. So in order to understand whether PCM1 help organize or regrow the microtubule, then we use a classical cell biology experiment, which typically we deplete all the microtubules within the cells with the drug. And then we like reculture those cells and to see how the microtubule really grow back. And this is in the control condition. You could see like in the later mid to like later stage cardiomyocytes, the microtubule started to regrow from the nuclear envelope. But however, in this PCM1 knockout, you don't see any of those. And you may see like a few of the dots inside of the cytosol that may be originated from the from the other like residue part of the centrosome, but not from the nuclear envelope, which suggesting that PCM1 help organize and grow the microtubule from the nuclear envelope during cardiomyocyte maturation. So then we started to perform the single cell transcriptomic sequencing, trying to understand how this really happens over time. And then we perform at 2 different time points. One is day 20 early stage, one is day 40 like mid stage. So as you can see, based on the transcriptomic expression, we can define the different clusters into whether they are like naive stage or they are in this intermediate levels of the maturation, or they show a structural mature stage, or like they show a electrophysiology maturation, and both at the day 20 and day 40. And to simplify our analysis here, and we use some mature sarcomere genes to quantify their sarcomere score. So which is really consistent with our cellular phenotype, we found in the early stage, in this knockout condition, the sarcomere score actually higher in control. And when you look at those sarcomere-related gene expression, is in KO, when you look at those gene expression, it also globally higher in the KO condition. But over time, when it getting into the middle stage, the KO showed a decreased level of the sarcomere genes. Here, I'm only showing you the sarcomere scores, but actually we look at the mitochondria, we look at the metabolism and other different things. We also saw a decrease in the day 40 cells.
The score is only relative to gene expression, not
to the
organization of the protein.
Exactly,
Okay.
exactly. So this is where we are getting into. And when we really look at the percentage of this structural mature populations, we also found at day 20 in KO condition, it increased, but by day 40, it decreased. So again, when we look at the geo terms, we found that the day 40 downregulated the muscle contraction and also muscle structural organization and some other cytoskeleton related genes. And another thing that really make us feel surprised is by day 40, the upregulation, we found like the mitochondrial genes and also ATP related electron transport is actually increased in the KO condition. And this increase is also happened in the day 20 knockout cardiomyocytes. So which is not like really consistent with our results. So therefore, because this is only the transcriptional level, so we started to look at what mitochondria really happens in the PCM1 knockout cells. And indeed, here we stained the mitochondria and also microtubule at the same time. So microtubule here in red and mitochondria in white color. In the PCM1 knockout, you do see an increased mitochondrial mass. But however, in all of the cells, it starts to show this kind of like toward one side accumulated, bundled together structure. And then we started to see what really happens to those mitochondria. And then we performed this live imaging. And as you can see, if you like really focus on this white boxes over there in the control condition, you will see this white color mitochondria is actually migrating on the microtubule. But however, in the KO condition, you will see the wiggle around of the mitochondria, but they are not actually really moving actively. So then with that, when we really look at the middle stage to the later stage, you will like really appreciate to see like in the control condition, the mitochondria started to show typical this like a connected structure. But however, in the PCM1 knockout, you will see like a lot of mitochondria still there, but they are like fully disrupted from the functionally. So over that, let me like briefly summarize what our results really showed. Like in this control cardiomyocytes during this maturation, it really initiated when those per-proliferative cardiomyocytes exit the cell cycle, with the centrosome started to disassemble, and part of the centrosome proteins they translocate to the nuclear envelope, and which facilitate the reorganization of the microtubules. And those microtubules, they are crucial to redistribute the organelles or help reorganize the sarcomere and for its maturation. When the PCM1 is gone, you will see maybe the fission and fusion of the mitochondria is disrupted, and also the sarcomere structure is also disrupted. And so overall, I hope our data really convince you that this perinuclear organization of microtubules is a central cellular mechanism that coordinate mitochondrial dynamics and also sarcomere maturation. So with that, I'm really thinking about what this data really implies and what from another level to envision the whole picture about pro-proliferative to the maturation transition. So I hope I convince you that from this fetal stage cardiomyocytes to the adult cardiomyocytes, it's actually a phase change. It's not simply a quantitative increase of the organelles or sarcomere or the cell size. But how does this happen? Because in the field, we are all focusing on what transcriptional regulation really helps this. And there are some transcriptional factors has been identified, and also signaling pathways like HIPPO, like HOXP2, HOXP1, and some other transcription factors. They all show a certain increase of the maturation when those transcription factors are overexpressed. However, what we show is that may not be sufficient. It also required a settled architecture foundation. So this is almost like transcriptional level provides the resources, and the settled architecture provide the foundation, right? The structure, so that the cells can really go from, if you really think this is a one-bedroom apartment, and then one is getting to the adult cardiomyocytes, it's kind of like a skyscraper, right? It requires a fundamental change of the structure.
Do you see myocyte proliferation change in your PCM1 knockout?
Yes, it decreased
Okay.
in the later stage,
Okay.
right? So how
even more so than fetal to adult in the control?
Fetal to adult in the control?
Yeah.
Yes.
Okay.
Yes, exactly. And so then why this is important, right? I feel this is relevant to multiple questions. And the two most questions that I constantly ask our lab or myself is why adult cardiomyocytes rarely divide? When you really look at the structure over here and how the centrosome really totally massively remodeled. So you can imagine this is really hard to disrupt everything and then go back to proliferate and then rebuild everything. And then another question is why iPSC cardiomyocytes cannot fully mature in vitro with our current protocol, right? One, we do see the PCM1 disassemble in the iPSC cardiomyocytes, but we feel that is the first step toward the maturation. There should be more things happening at the same time.
Have we tried overexpressing?
We are working on it right now. Yeah, we are hoping that can really help it. Yes, exactly. So I know from the whole talk, you have been keep thinking about how this is really relevant to the heart disease. So I'll briefly talk a little bit over here. So the centrosome and microtubule has been broadly associated with different types of disease, and mostly broadly studied in cancer, because as you can imagine, the dysregulation of the centrosome and more duplication of the centrosome can really cause the cell cycle problems. So it directly relevant to the cancer. And from another perspective, it can also cause the congenital defect, like usually it's multi-organ defect. For example, like microcephaly and the PCNT, the one I showed earlier, that could cause the microcephaly and also accompanied with the congenital heart defect. And some also the ciliopathies. And here it may be a long story, but like in short, the centrosome and CLU, they share the similar structures and the same proteins. So that's why they share some similar disease. And the ciliopathy has been found to broadly associate with the congenital heart defect, as I'm writing over here, the heterotaxy and even BAV and different types of disease. And it's also not only in cardiomyocytes, but also in endocardial cells, endothelial cells, and different other cell types. And some other research also found it can cause the familial dilated cardiomyopathy and also related to the heart failure with the abnormal microtubule structure. So I'm only stretch a little bit of some of the data we have over here. And over the past few years, like the researchers started to find that microtubule is totally messed up in the heart failure patient. And here is showing you the image of the microtubule, both the total microtubule and post-translational modified microtubule. And this is the control healthy heart, and this is failing heart. You see the dramatic increase of the microtubule. And from the bulk RNA-seq, from multiple data sets, they also found the same thing. The centrosome and the nuclear peripheral and the microtubule organization center all dysregulated. So, but however, what are the mechanism, what are the molecules that's really responsible for this is really not known. So our medical student, Anna Turtle, during the past summer, started this small project by looking at centrosome and microtubule in the patient samples. And due to the limited resources, so far she collected 1 control sample and 2 heart failure samples. And those samples are all the HFrEF. And so we examined the PCM1. And from the images you are seeing over here, compared to control versus the heart failure samples, I believe you can appreciate a dramatic increase of the PCM1 in the heart failure samples. However, we failed to really have a good quality of the microtubule staining, but this data kind of like suggests that maybe PCM1 is getting involved in that increased and messy microtubule organization in those heart failure samples. And we are still trying to work on like different disease types, maybe like HCM or like DCM. So that's overall our results. And those 2 projects, as I mentioned, led by Suki and also Anna. And we have like multiple collaborators who have been contributed to these projects. And finally, our funding resources. Thank you all. And I'd like to take any questions.
Any human mutations in PCM1 or any of the
There's some SNPs has been like associated with the neuron degenerative disease and also the arrhythmia.
Okay.
Yeah, so we are like really looking more into that part.
Yeah.
Hi, fascinating talk and really intriguing findings. I was wondering how PC1 expression, either transcriptionally or protein level expression, sort of what is its trajectory during cardiac development? If we have any insight into that, and like how does that correlate with your studies? You know, is there a temporal correlation, for example?
Yeah, from the transcriptional level. So we have found that PCM1 expression increased from the embryonic to the postnatal stage. So so far we are not quite sure about how it really like changed during the heart failure. That's probably something we should examine in the heart disease. And I'm guessing for the protein level, it should also be consistent, but so far we don't have that results.
Yeah, just
did I answer your question?
Yeah. Just to kind of tie that together, because I think one of the issues is limitations with knockout studies and overexpression studies is that we don't really have temporal control whether or not this is something that you want to think about in a mouse that you could, for example, have an inducible Cree that then can, activate or reduce PC1 and then see, kind of how that affects development.
Yeah. We are currently working on the conditional knockout, and we also have the global knockout PCM1 mice. And there's also some data I didn't show today that we also did the inducible CRISPR-Cas9 knockout of PCM1 only in the iPSC cardiomyocytes. And we found is similar results as shown from over here, the disruption of the sarcomere and less mature phenotype. Okay, all right. So thank you everyone for coming. And like if there's in the future any interest for collaboration or like any resources about the tissue availability, I'm really appreciate and happy to know. And for our next meeting, so I guess we already have scheduled. So, all right, if there's no question, and thank you for the seminar from Jingwu today. I'll see you next time. Bye.